Looks good.
Let me just go to the beginning. Is Bruce going to test his also? He already did.
I'm good. I did it.
Okay. You want me to get out or should I leave it here?
Maybe get out for now, and then you'll be the first speaker.
All right.
Yeah. Was there any trouble logging in with the link? Because I had some issue on my end.
I got on just fine.
Oh, okay.
Me too. No problems.
Cool. Okay. Yeah, thanks guys for joining.
Thank you for inviting.
Yeah, seriously.
Are you going to SFN?
I haven't made a decision yet. Still got a little bit of time, right? Is it next week?
Yeah, I have no excuses. It's in San Diego, so I have to go. But I literally registered 2 minutes ago.
Oh, okay.
It's only a 2-hour drive for me. Yeah, I still am having trouble committing. Are you going, Aaron?
I cannot go this year, but next time.
That's what better to do.
No.
It's a shame. I started a wine collective at Scripps, so I was eager to repay you for all the wine that you gave us at the conference.
Yeah.
There'll be other chances for that.
Yeah.
Aaron.
Hi, Long. Do you have any trouble logging in, Long?
Yeah, I'm fine now. I'm getting a new network. Hi,
Okay.
Bruce.
Hey.
Hi.
Hi. Your first name, Giordano?
Giordano, yes.
Giordano. Yeah, great. I saw you in Darwin's lab, right?
Yes.
Before. So I work in the ground floor in the Pacific for 6 years.
Nice.
I was with Anima Ghosh.
Oh, very nice.
Yeah, Dr. Roche and many more. So I'm very familiar with Nick Spitzer, Nick Darwin, me, many colleagues, things out there.
Yeah. Nick is giving his Festschreit, so his 50 years, 50-year UCSD party, but he's not retiring. He's just giving a party.
Great. Yeah, he's awesome. He can be on and on.
Yeah.
Yeah, I miss him. And Nick visited China a few times back in. We come to Suzhou and Shanghai for our institute interview, institute reviewing us for tenureship.
Yeah. I remember Darwin going too and saying the first time he came back, he said that he was blown away by the level of the science. He was really impressed. Yeah.
So keep working. Yeah. I miss San Diego. I've been there.
Are you coming to SFN?
Not this year. I've been there like 2016. I went to the Pack Hall, actually. I particularly drive to Pack Hall, find out the whole field is being building some new building, right?
Yeah.
Yeah,
that's done now. And there is so many other buildings. They're building like crazy.
Okay.
Yeah.
People used to play football on the field.
Yeah, they kept that actually. They kept, but they made it artificial grasses and proper.
Great.
Yeah, but they built the Tata building just next to it.
Oh, okay. It's a new building then.
Yeah.
Great. Yeah, excuse me. I'm trying out my new headphone.
Oh, sounds good.
Whether it's working or not. Yeah, sorry.
Is Dion coming today, Bruce?
Oh, I don't know. I haven't seen him today. I didn't ask him. Maybe. He did one of these, right?
Yeah.
Okay.
Yeah.
Hey, Bruce.
Who's that? Oh, hey. It's been a little while.
Unbelievable. We're stuck here. Can't go anywhere. The only person I can see is Aaron.
Yeah. Well, I feel like I just saw you a couple hours ago, Yellen.
Yeah. Yeah, we hang out a lot together.
Yeah. We had a crazy Monday night party last night.
Oh,
Really?
Nice.
Only 9? Wow.
Wow, some dragon bed.
Yeah. My kids are out of town for like a school trip for 3 days, so I had nothing to do.
yeah, party.
We had a Monday night party.
Looking
little
forward.
catch- How's Justin doing, Bruce?
Oh, I think he's doing all right. I just got an email from someone in his lab wanting me to be on their thesis committee. So
Nice. It seems like Giordano, someone at Scripps wins Nobel Prize every year.
Yeah, and Barry got his second Nobel Prize. That is actually pretty spectacular. It's gonna be hard to beat. Yeah, and apparently during his sort of celebration, one of the stories was from our vice president that the day of the award, he got into Barry's office and said, you know, there are like 50 journalists out there. We need to talk about it. And Barry doesn't care at all about all this awards and stuff. He was like, have you heard of this newest, chemistry thing that they just found? And just start drawing on the drawing board. Just like, it's just so refreshing, that people like that still exist.
Yeah, people are still crazy about the price, right? In the US, especially in China, China gonna invite all kinds of Nobel laureates and come here to give
So
all
how did
kinds of
they-
lectures.
Did
Yeah.
they have to quarantine? I saw like there's like 20.
You get some special quarantine, I bet, like 3 days quarantine, something.
Yeah, I wonder because Rothman is there, Sheckman. I can't imagine that they would quarantine for
Yeah, I
days.
bet no more than 3 days, and you can free to go.
Yeah. How do you get that? I have to figure out that.
And the Nobel Prize, right?
Yeah.
A special protocol for all kind of things.
Yeah, just get Nobel Prize.
Yeah. That might be an easier way to get out of quarantine these days.
Right.
Very, very constructive suggestion.
Yeah.
So get started, Aaron.
Yeah. Great. Okay. Hi, everyone. Another NeuroZoom week. Next week is the Society for Neuroscience meeting. So we'll take 1 week off, but then we'll be back at it the following week, and just today's in Asia, it's an hour later. So in America, it doesn't feel any different. So just note the new time, but we'll be able to get right into our first set of talks now. Zilong can introduce Giordano, and please make sure to tell your colleagues if they'd like to give a talk to let us know. We're always looking for good speakers, and you can check out the YouTube channel where the talks are streamed live. You can subscribe to that or just check it out. Okay, Zilong.
Sure. It's my great pleasure to introduce Dr. Giordano Lippi. Giordano now is assistant professor in Scripps Research Institute in San Diego. So Giordano has his bachelor degree and a PhD in Italy in the University of Modena. So after that, he briefly worked in UK, I believe. And then ever since he stayed in beautiful San Diego, okay, postdoc with Professor Darwin Berg. And when he started to study the amazing role in a series of microRNA in neurodevelopment, and after postdoc, he started his own lab in Scripps Research Institute, as well as the joint position in UCSD. So today, Giordano will talk about his latest in his own lab, inducible and reversible microRNA loss function reviews critical developmental windows. Okay, welcome, Giordano. Take it away.
All right, let me share my screen, start the presentation. The usual check,
Sure.
It's good. Okay.
Yeah, good.
All right. So as mentioned in my lab, we're very excited about non-coding RNAs and microRNAs in particular. And the reason why we're so excited, okay, let me get my laser pointer here, is graphs like this one. And this shows the relationship between biological complexity as increasing number of different cell types and the number of protein coding genes. And so each organism is a square. You see bacteria down here, for example. And you might have already noticed that there is a problem with this graph. There is, since the dawn of multicellularity with this amoeba, all the way to humans, it doesn't really seem to be an increase in the number of protein coding genes. And so how is this possible? Where is the genetic information that is needed to achieve such a big leap in complexity? And the answer is most likely here. If you now plot for the same organisms, the number of non-coding genes, where you see this beautiful linear relationship. And so what this is telling you is that to achieve greater complexity, evolution did not add more building blocks, but instead mechanisms regulating gene expression in space and time. And this expansion of the non-coding RNA repertoire is particularly dramatic if you look at our favorite class of non-coding RNA, that is microRNAs, as I mentioned before. And this graph shows you the new microRNA families that emerged in the last 400 million years in evolution. And you can see that compared to simpler organisms appear, mammals and primates in particular saw really an explosion in the number of new microRNA families. A lot of these families are enriching the brain. And so what this is suggesting is that microRNAs are necessary to assemble and maintain a very complex organ like the mammalian brain. So a quick reminder, what are microRNAs? This is normal mechanisms of translation of an RNA. MicroRNAs are loaded in the Argonaute complex, or AGO, and they recognize their targets based on sequence complementarity to a microRNA recognition element, or MRE. And when this interaction occurs, it recruits a family of protein called TNRC6. Remember this TNRC6 family of protein because it's going to come back multiple times. And this is really the effector of microRNA function. And without getting into many details, all you need to know is when this interaction occurs, you have repression of gene expression for that specific target. And this just to show you sort of that, it was a cartoon that sort of the real relationships. So this is an RNA, this squiggly line, this is a ribosome, and this tiny thing, Argonaute, with a microRNA loaded inside, is going to do all the functions that I'm going to describe you today. All right, so of course, this is one microRNA, but the reality is there is hundreds of microRNAs expressed at medium to high level at any given time in any cell type. And these microRNA form a network, a microRNA target network. And really, we should be thinking about microRNAs as a network. And this is a sort of a cartoon, a scheme of how that network looks like. Numerically, it's actually pretty accurate. We've done a lot of mapping on these networks. and circles of microRNAs, squares of the targets, the size of either. It tells you how abundant they are. The line tells you what kind of repression is occurring. And the thicker the line, the stronger the repression. And what you can observe is these networks have special characteristics. For example, there is a lot of redundancy. Multiple microRNAs are converging on the same targets, and also they're not homogeneous. So the periphery of the network, there is very little going on. At the center, there is a lot going on. Are these networks important for brain development? Well, of course they are, otherwise I wouldn't be here talking to you. If you remove this biogenesis enzyme called dicer that converts an inactive precursor microRNA into the active mature form of the microRNA, you cannot form mature microRNAs anymore. If you do it in cortical progenitors, this is a normal brain at P0. Here is when you knock out dicer, you can see very dramatic phenotype, drastic decrease in the size of cortex, and drastic decrease in the size of the hippocampus. So what this is telling us is that it's not just important, that as important as turning on genes is when you turn them off and how much of that gene you're expressing. Okay, so based on this sort of early exciting data, I would say that sort of the most important questions that we should have answered as a field is where are microRNAs important? So are microRNAs roles universal or specific to certain cell types, and when? So which developmental stages during aging or maybe diseases? So both of these are very important questions. And then the how. So what are the molecular mechanisms the microRNAs use to regulate the development of the brain? And I'm going to argue that as a field we still know relatively little. And this is both for technical and conceptual limitations. And today I'm going to really focus on the technical limitations and share with you some new data that I'm very excited about, and hopefully I'm going to get some feedback from you guys. All right, let's get into the technical limitations of what we've been doing so far. So this is the developmental timeline of a pyramidal neuron of the cortex, and you can see neurogenesis in the embryo and then differentiation once the animal is born. And strong phenotypes that I showed you before with the dicer knockout was done with this EMX1 Cre line, in which basically the Cre kicks in around E9. And you can see from the title of the paper itself that what they found is an effect on differentiation, but not on neurogenesis, okay? So, one can conclude, like the authors concluded, that microRNAs have stage-specific roles. So they're not important for this, but they're important for that. Or maybe there is a delay in the onset of the phenotype. Even worse, if you look at this beautiful cell type of the cerebellum, Purkinje cell, that is going to be the topic of the rest of my presentation. If you use an L7 Cre line that is specific for this cell type, but kicks in at P4 so posterior day 4, it has absolutely no effect on the very prolonged differentiation of this cell type. It takes up to 4 weeks for these dendrites to fully develop. And you need to wait 8 to 10 weeks to see a neurodegeneration phenotype. And so the conclusion could be that either microRNA roles are cell type specific, so they're not important for Purkinje cells during development, or that there is a very long delay before the onset of this phenotype. And we have quite a bit of data in the lab to show that the second hypothesis, the one of the delay, is really what's going on. And this is because the half-life of microRNAs can be extremely long, in particular in postmitotic neurons. And this is because both ends of the microRNA are locked into the argonal complex, and so it's very easy to degrade it. So if this is true, it also means that microRNA roles are being largely underestimated by the last 15 years of research in this field. All right, so we need something better and faster to be able to deal with the quick timeline of development of the mouse brain. As I told you, we really cannot do anything about the levels of mature microRNAs, but maybe what we can do is we can mess with the downstream effectors of microRNA function, for example, TNC6. And we were lucky that Günter Meisser's lab developed a peptide called T6P that is basically this portion of TNC6 that has high affinity for argonal proteins, and they developed it to be able to pull down microRNA target interactions, basically map microRNA target interactions. What we reasoned is if we express enough of T6P, this might act as a dominant negative, and prevent TNFC from binding to that pocket, and maybe de-repress all microRNA targets much more rapidly than a genetic approach like dicer knockout. Does it work? All right, so again, pay a little attention here because this tool is going to come back multiple times. This is what we call the microRNA sensor. Has two fluorescent proteins, one going this way, blue, and one going that way, cherry or red. And the cherry is regulated by a very abundant microRNA 124. So it's repressed by 124. We expect to have high levels of BFP and low levels of cherry. Then we use another virus in which we are expressing that peptide that I just told you about, that we fuse it with YFP. And then as a control, we use this mutated peptide, in which basically we mutated every one of 5 tryptophans that are very important for the binding of TCSP to the argonauts protein. And this is what we see. So this is normal condition. You see these 2 neurons were transduced with a sensor. So you see high levels of BFP and very low level of cherry, because cherry is repressed by microRNA 124. And when we deliver this peptide TCSP and block microRNA function, we see a very nice increase in levels of cherry, and this is quantified here. And as you can see, we are not that far when we use TCSP from this internal control, in which we basically mutated all the bases in the seeds that are important for 124 to bind to the MRE. So basically, this is when cherry is not repressed by microRNA 124 anymore. Is there a functional consequence to blocking microRNAs? Indeed, there is. So you can see here in pyramidal neurons in culture, there's a normal pyramidal neuron with beautiful dendritic architecture, and that is really, really stunned when we use TCSP. This is an effect we already know is linked to microRNA function. So this was very reassuring to see. And so we're very excited because, of course, this is a peptide. You can express it very rapidly, and also because you can do some vector and protein engineering and make it satellite specific, make it inducible, make it reversible, and restrict it to different compartments of the cell. And so basically achieving extremely high spatial and temporal resolution of microRNA loss of function, something that we have never been able to do before, and that is going to allow us to really address that where and when questions that I was telling you at the beginning. All right, so the lowest possible hanging fruit that there was in the field is the development of Purkinje cells. As I told you, what the field believes so far is that if you knock out dicer with Del7 increase, starting at P4, you get absolutely no effect on this protracted development of the dendritic arbor, okay? This takes 3 to 4 weeks, so it's a long development, but absolutely no effect. So this was our benchmark. What we did is we cloned the exact same promoter that is used for this Cre line, this L7 promoter. This will, if we inject this virus in the ventricle, transduces exclusively Purkinje cells. You can see here, this is a sagittal section of the cerebellum, and zoomed in. You can see these are sort of very beautifully labeled Purkinje cells of multiple ages, and this is with our control that is basically a YFP protein with a little peptide attached to it. And so we wanted to know, does it block microRNA function in vivo? And so we use our 124 sensor again. As you remember, what we expect to see if microRNAs are active is high levels of BFP and low cherry. And this is what happens with our control. But when we block it with TCSP, what we see is high levels of BFP and high levels of cherry, and this is quantified here. And I hope you can see right away that something is very wrong here. Like these cell bodies look very different, right? And in fact, the difference between the two animals was sort of visible to the naked eye. You can see the cerebellum is much smaller. This is 3 weeks. At 3 weeks of postnatal life, we cut it, it's much smaller. The phenotypes emerge as early as 3 days after this promoter kicks in, and it becomes increasingly larger. And the phenotype was so severe that we thought, well, maybe this is enough to affect behaviors that have been linked to the function of Purkinje cells. So the most obvious one is motor coordination. So we did the accelerated rotary test, in which basically the animals have to learn to stay on top of this rotating rod. And you can see normal animals are pretty good at it. But the animals in which we blocked microRNA with TCSP are really not capable of doing this. And then we did another test. Recently, there has been a link between sociability and Purkinje cells. In fact, in autistic patients, the most frequent defect is a decrease in the number of Purkinje cells. And so with this 3-chamber sociability test, what you normally expect is a mouse prefers to interact with a new animal instead of a new object. You can see this heat map here, likes to stay here more than there. And when you block microRNA with DXSP, what you see is that preference is pretty much lost. And this is quantified here. All right, so strong phenotype, this is sort of a big novelty in the field. We're very excited. But I think this is also an opportunity for us to really get into some cellular mechanisms of microRNA function. As I was telling you before, there are a few of the things that are disruptive of microRNAs, and we decided to focus on 2. One is the actual dendritic arborization that takes roughly 3 weeks to go from this sort of fusiform form to this beautiful arborized structure. We picked dendritic arborization because the beauty of the cell type that exists in a single plane, so you can really reconstruct the entire dendritic arbor relatively easily, and also because it's a cell autonomous and intrinsic program. So this is the cell type in which, the only cell type in which we're blocking microRNA. So we expect this to be severely affected. And then we picked another sort of developmental event that is happening, that is the innervation by the climbing fibers that are coming from the inferior olives. These are excitatory connections, VGLUT2 positive. And as you can see, it's called climbing fibers because there is a translocation of the axon along the dendritic tree that happens roughly sort of peaking in the 2nd and 3rd week of postnatal life. All right, so what happens when we block microRNAs? This is a normal Purkinje cells, beautiful architecture. As you can see, very, very reduced complexity of the arbor that is quantified here. That is another phenotype that we see, sort of architectural phenotype that we see, is a stunted growth of the sort of a reduced height of the dendritic tree, and this is quantified here. And then what we see is also an effect on these VGLUT2 puncta that are basically the climbing fiber. You can actually, this is basically a dendrite that you can track just by looking at the dots of the VGLUT2 that is going there. This is also severely affected. So both of these phenotypes are severely affected by the lack of microRNAs, and this is exciting for us because we can start asking questions that nobody has asked before. And in particular, what we can start asking is, are there critical developmental windows in which microRNAs affect maybe one and not the other of these 2 phenomena that I just showed you? All right, so to be able to do that, not only we need something that blocks microRNA fast, but we also need a reversible version of the same tool. And so we added a diagram moiety that is called DHFR. When this is produced, it is degraded very rapidly, so it never accumulates. But if you add TMP, what it does, it binds to this DHFR and stabilizes a molecule, so you can very rapidly accumulate DCSP because RNA is already present, okay? And so what this does, basically, you can induce the repression very rapidly. And the cool thing is that, as you can see here, so this is before we give TMP, there is very little green, if no green at all. And you can see there are 2 cells here that receive the sensor in vivo, and mCherry is very low, as we expect, because microRNA function is still active. After 18 hours to giving TMP, there is bright expression of DCSP, and you can see the repression of mCherry, so microRNA function is blocked. And you wait all the way to 36 hours, and DCSP is gone, and also microRNAs came back to normal, right? And this is quantified here. You can see nice induction of the fluorescent DCSP with a little delay for the 124 sensor, because the protein, of course, has to be produced. So we have a nice sort of 12-hour window in which we can turn on and then turn off microRNA function. And the first experiment that we decided to do was to look at those 2 developmental events that I showed you before, and basically give TMP to the animal every 12 hours for a week. And we decided to block microRNA's function for week 1 or week 2 or week 3 or week 4, and then collect all the animals at the same age, that is P28. And the hope was that we were able to find a critical developmental window in which one of the 2 developmental events was affected, but not the other one. Okay, so we were finally able to sort of separate these developmental events and how microRNAs regulate them. All right, so this is the data. If we block, we're looking at the size of the cerebellum. That is sort of an indication of the dendritic arbor. You can see week 1 and week 2 induce the phenotype, but week 3 and week 4 don't. And this is the same if we look at the height of the dendritic arbor. Week 1 and week 2 induce a phenotype, but week 3 and week 4 don't. And this is probably one of the most exciting piece of data that we've had in the lab since my lab started. If you now look at the climbing fibers, what you see is that week 1 and 2 have no effect. But if you look at week 3, there is an effect. And this is particularly exciting to me because week 1 and 2, as you can see here, they have a smaller dendritic arbor. So what this is telling us is that just by reducing the scaffold in which this climbing fiber is growing is not sufficient to induce the phenotype. What you really need to do is to mess with gene expression at a time in which there is a sort of interaction between cell intrinsic factors and sort of factors from another cell type that is trying to synapse on it. All right, so now that we have this critical developmental window in which we can block microRNA, what we want to do is trying to get a mechanism. And to get a mechanism, what we do is basically map microRNA target interactions and then see if we can find vulnerable nodes of this microRNA target network. For example, abundant microRNAs. That's sort of the classic way that we've been studying these microRNA target networks. And so the first thing that we need to do is actually map those microRNA target interactions, and we use this technology called CLIP-seq of Argonaut2. CLIP stands for cross-linking immunoprecipitation of Argonaut2. We have a conditional Argo2 that we can express exclusively in Purkinje cells. We freeze the microRNA target interaction with UV light. We digest the part of the RNA that are not protected by Argonaut proteins, and then we IP for the tag. So only interactions that were occurring in Purkinje cells. And we send everything for sequencing. We get peaks, and those peaks tells us that the interaction was very frequent. And now we developed an extra part of the technology in which we link the microRNA and the target so that we actually know which microRNA was binding to that specific target in what they call microRNA target chimeras. And this is real data. So this is showing you there are targets at the top, there is microRNAs at the bottom. This is what we call a bipartite network. Basically, every microRNA target interaction is depicted here. This is roughly 10,000 genes up here. And so now that we have this map, we can really start trying to figure out which are, for example, the microRNAs that are doing most of the job. And I'm showing you this heat map here, in which basically what you see is that the sort of classic microRNAs you would expect, like 124, 138, the LS7s are highly expressed in this cell type during development. But what we're very interested in is a microRNA like this one, microRNA 206. You can see here, if you plot the abundance of microRNA that come down from these CLIP experiments in pyramidal neurons versus Purkinje cells, the majority of microRNAs are very similar in levels. But there are a few that are enriched in pyramidal neurons, and this guy in particular is very highly expressed in Purkinje cells, but completely absent for pyramidal neurons. So if there is any sort of blueprint difference in the architecture or in the function of this cell type, this microRNA and a few others might be very important for it. So we've designed sponges to be able to get rid of those microRNAs. And then something I've not talked about at all, we're also thinking about microRNA hotspots, so targets that are very heavily repressed by microRNAs. And we've developed an RNA CRISPR system that, again, I'm not going to talk about today. It would take another probably 20 minutes. All right. So I hope I convinced you that we have developed tools that for the first time give us really a possibility to address that where and when microRNAs are important during the development of the mouse brain and dissect microRNA roles at unprecedented spatial and temporal resolution. And we're working pretty hard to be able to also do it in subcellular localization, axons versus dendrites versus spines. These are also questions that we're very interested in. And the mechanisms I briefly touch upon, we're doing CLIP, we're doing sponges and RNA CRISPR systems. And this is just to say that this work was spearheaded by the first graduate student in my lab, Nori, and in collaborations with quite a few members of the lab and other labs, the McRae lab and the Yale lab. And this is a funding that allow us to do this work. And any questions?
Great, thanks, Giordano. Beautiful work. I may have a quick question first. So, beautiful work reminds me one of my first work in my own lab is finding out the methyl CpG binding protein MECP2 regulate the nuclear microRNA processing. So back then we found that microRNA seems to play some role in the dendritic development as well as the synaptic development. But in terms of cerebellum, it's very amazing to have a so you have a wider role in the differentiation. Do you think is there in terms of indeed it has a differentiation of your stem cell related to the cerebellum? So or they are purely due to the lack of cell number, or your cells just stuck into the neural stem cell phase that couldn't differentiate?
Yeah. So the earlier you go with dicer knockout, right?
Mm-hmm.
If you're able to do dicer knockout very early, you see, right? For example, a paper came out last year from the Borel lab that shows that if you actually go much earlier, like age 7, you do see an effect on neurogenesis, right? So my prediction today is that every single developmental thing that you study, microRNAs have important roles. It's just they are too prevalent, too highly conserved. There is just too much going on for not to have roles. Now, you briefly mentioned what you were talking about. So I think that the beauty of this tool is that as long as you have a way of expressing it, so if you have a promoter, or if you can turn it on rapidly, you will find out exactly. It doesn't matter how quick that process is. You will find out if microRNAs matter or not. Now, you briefly mentioned what the things that you're doing in the lab, nuclear processing of microRNAs. As you know, there is also quite a bit of work that in the last few years has been done on roles of microRNAs, so mature microRNAs in the nucleus, right? So there is this new literature showing the microRNAs have roles in the nucleus. So one of the experiments that I didn't show, but that we've done is basically we use a nuclear exporting sequence for TCSP, or a nuclear localization sequence for TCSP. And what we see is the majority of what we see is actually classical cytoplasmic roles for microRNAs. We have not seen much, at least for the phenotypes that we're looking at for nuclear roles. But I think this is why this tool is exciting, is because there are all these questions that are sort of open, and there are people that think that microRNAs do this, maybe they don't do that. And this is a way to finally say, this is what they do, this is when they do it, and there shouldn't be any more open questions, hopefully soon.
Great, thanks. Okay, any more questions?
Okay, should we go to the next one? Too long?
Oh, great. Okay, thanks, Giordano. Great.
Thank you very much.
Great, thank you.
Right. Thanks so much, Giordano. And now our next speaker is Dr. Bruce Herring, who is coming from University of Southern California. And Bruce received his undergraduate degree in Texas at University of Texas at Austin. He then did a PhD at University of Chicago, where he had very important studies about the role of anesthetics and how they work on the central nervous system. And he discovered a role for these in acting directly on neurotransmitter release, and this provided insight into their mechanism of action. He then did his postdoctoral fellowship at UCSF with Dr. Roger Nicole, and here he studied LTP, and it was long known that LTP is regulated by CAM kinase 2, but the targets of CAM kinase 2 weren't completely resolved. And here he discovered 2 proteins, calirin and trio, as targets of CAMK2, and they're phosphorylated, and it's required to regulate AMPA receptors during LTP. He also studied other adaptor proteins and how they can regulate neuronal AMPA receptors. In 2016, he moved to USC, started his own laboratory in the neurobiology department, where he's continuing to make great progress, and he'll share his latest results with us. Thanks, Bruce.
Thank you all for being here. Thank you, Aaron, for inviting me. So the title of my talk today is Understanding Synaptic Dysfunction and Schizophrenia. So I'll start with sort of a very broad background slide on schizophrenia. Schizophrenia is a debilitating neuropsychiatric disorder. It affects around 20 million people worldwide. It's characterized by auditory hallucinations, delusions, flat affect, poor executive function, and deficits in memory. And we know exceptionally little about the cellular molecular underpinnings of this disorder. One thing that we do know about schizophrenia, based on both clinical and preclinical data, is that glutamatergic neurotransmission is elevated in the hippocampus in the early stages of this disease, on the prodromal stages, and that these elevations in hippocampal glutamatergic neurotransmission positively correlate with schizophrenia progression and symptom severity. And it has been proposed that this hippocampal pathology acts as a seed for schizophrenia development in the brain by causing functional alterations in extrahippocampal structures like subcortical dopamine centers, where elevated glutamate coming from the hippocampus is believed to produce elevated dopamine release in the brain, which is another hallmark of the disorder. But what we don't know, again, are the cellular molecular mechanisms that are causing elevated glutamatergic neurotransmission in the hippocampus. And that's sort of a question that my lab has been interested in. And to try to answer these questions, we've turned to human genetics. So this slide represents our current understanding of the genetics of schizophrenia. This is very recent data that comes from Schizophrenia Exome Meta-Analysis Consortium, or SCHEMA Consortium. This is a partnership between human geneticists at MIT and Harvard. And so each one of these green and red dots represents a genetic perturbation that has achieved whole genome significance for the disorder. And so for the purposes of this talk, I'm going to focus primarily on 3q29 microdeletion mutations. So individuals that harbor this microdeletion mutation, their likelihood of developing schizophrenia increases by over 40-fold. This is the largest increase in risk produced by any genetic perturbation that we are currently aware of. Mice have been made that harbor this microdeletion mutation and reduced protein expression of 6 genes in the microdeletion locus has been shown in this preclinical model. These are those 6 genes, but of these 6 genes, the gene DLG1, which encodes the protein SAP97, is generally considered to be the favorite as a disease driver, given that individuals with schizophrenia have been identified that harbor missense mutations that are clustered specifically within SAP97's PDZ2 domain. So what do we know about SAP97? Well, it is a member of the McGuck protein family. Other members of this family include PSD95, PSD93, and SAP102. We know quite a bit about PSD95, PSD93, and SAP102. What they do is they sit at the postsynaptic side of glutamatergic synapses, where they act as scaffolding proteins that hold AMPA and NMDA receptors in place so that they can be efficiently activated when glutamate is released from the presynaptic neuron. And in the event that you get rid of PSD95, PSD93, and SAP102, this causes AMPA and NMDA receptors to fall out of glutamatergic synapses. This has been shown by many groups and is evidenced by synaptic AMP currents and synaptic NMDA currents, the amplitudes of these currents being decreased significantly when PSD95, PSD93, and SAP102 are eliminated in neurons. So SAP97 is very much the odd duck in this protein family. So, and this is due to the fact that previous attempts to try to identify a role for SAP97 in glutamatergic synapse regulation. Many of these studies have been performed looking at hippocampal CA3, CA1 synapses. And so these are the most heavily studied glutamatergic synapse in the brain here in the hippocampus, and are generally used as sort of a model for all glutamatergic synapses in the brain. And if you get rid of SAP97 and these hippocampal CA1 pyramidal neurons, the postsynaptic neurons, the synapse, there is no effect on glutamatergic synapse function. Synaptic AMP receptor currents are normal in CA1 pyramidal neurons. Synaptic NMDA currents are normal. This is work from my lab using an RNAi against SAP97, but this was shown previously back in 2009 by Roger Nichols' group, who basically again found no change in glutamatergic synapse function in these CA1 pyramidal neurons. And this has led groups to sort of generally conclude that deleting SAP97 has no effect on synaptic transmission. So this was frustrating to us for a number of reasons. One, given SAP97's growing implication in schizophrenia. The fact that SAP97 is so closely related to very important glutamatergic synapse regulatory proteins, and the fact that schizophrenia is generally considered to be a synaptic disorder. And so this led my lab to ask ourselves, are we looking in the right place for synaptic role for SAP97? So as it turns out, there's another subregion of the hippocampus called the dentate gyrus, and the dysfunction of the dentate gyrus is increasingly implicated in schizophrenia development, with deficits in dentate gyral function being believed to produce many of the behavioral phenotypes that are observed in individuals with schizophrenia. So we thought we would do a very simple experiment, and that is to simply look at where SAP97 is in the hippocampus. And so we took rat brain slices and took an antibody against SAP97 and just looked at where endogenous SAP97 is in the hippocampus. And what we found was pretty exciting to us. So we find that SAP97 is in the cell bodies of CA1 pyramidal neurons, but is absent in their dendrites. Okay, so this is where the glutamatergic synapses are in CA1 pyramidal neurons. So that would explain why people have had difficulty trying to find a role for SAP97 synaptic regulation in synapses in these neurons, because it's not there. But in dentate granule neurons, in the dentate gyrus, you see something very, very, very different. And so their dendrites are full of SAP97. So we are very interested to know whether or not SAP97 might be playing a role at the synapses that form between entorhinal-cortical projection neurons, these axons that form glutamatergic synapses, onto these postsynaptic dentate granule neurons. I also say that the dentate gyrus is the input structure into the hippocampus. So as I showed you before, when you eliminate SAP97 expression in CA1 pyramidal neurons, you see no effect on glutamatergic synapse function. But when you instead get rid of SAP97 expression in these dentate granule neurons, you see something very, very, very different. You see this, as opposed to nothing, what you see in the CA1 pyramidal neurons, and dentate granule neurons, you see this marked increase in synaptic AMP receptor current amplitude. We see about a 4-fold increase in synaptic AMP receptor current amplitude. NMDA currents go up a little bit, but did not reach statistical significance in our assay. So what we think is going on here is that instead of sitting where PSD95, PSD93, and SAP102 sit in the postsynaptic side of glutamatergic synapses, what we think is going on here is that SAP97 actually resides in these extrasynaptic, perisynaptic regions of glutamatergic synapses in these dentate granule neurons, and that when you eliminate SAP97 expression, it causes this reserve pool of AMP receptors that SAP97 was holding away from the synapse to now allow them access to the postsynaptic side, leading to an increase in the number of AMP receptors that you have postsynaptically in these neurons. And this model is very consistent with some older work where SAP97 was recombinantly expressed in neurons, and electron microscopy was used to look at the subcellular localization of the SAP97. So this is a glutamatergic synapse. You have the presynaptic side of the synapse. You can see the glutamate-containing vesicles here. This is the postsynaptic side of the synapse. This is the PSD. This is where PSD95, PSD93, and SAP102 are, but SAP97 is not in the PSD. It seems to be, again, sort of localized to these perisynaptic or extrasynaptic regions. And so this is very much in line with our model. So another interesting thing about SAP97 is that it is the only MGUT protein that is able to bind directly to AMP receptors. And this interaction is made possible through the AMP receptor subunit GluA1, which has a intracellular C-terminal PDZ binding domain that binds to SAP97's PDZ2 domain. So this is very interesting because this is where you see this cluster of schizophrenia associated missense mutations is in SAP97's PDZ2 domain. So while we were, of course, very interested to know the impact that these missense mutations have on SAP97's ability to hold on to AMP receptors, the first thing that we wanted to convince ourselves of is that we wanted to know whether or not this interaction, that disruption of this interaction was really what was giving rise to the synaptic phenotype that we see in dentate granule neurons when we get rid of SAP97, because the PDZ domains can be promiscuous. So to convince ourselves that this interaction was relevant to the synaptic phenotype that we were seeing, we disrupted this interaction not by getting rid of SAP97, but instead by getting rid of GluA1's PDZ binding domain. And so what we did was to perform molecular replacement experiments where we got rid of endogenous GluA1 and put back, so we used an RNAi against GluA1 to get rid of endogenous GluA1 and put back GluA1 missing its PDZ binding domain. We did this both in CA1 pyramidal neurons as well as in dentate granule neurons. And what we find is that when we replace GluA1 with GluA1 missing its PDZ binding domain in CA1 pyramidal neurons, we find that synaptic AMP recurrents are unchanged. This was actually not that surprising to us. This was shown previously by another group. But when we instead replace GluA1 with GluA1 missing its PDZ binding domain and dentate granule neurons, we again see this very large increase in synaptic AMP receptor current amplitude, very much like what we see when we get rid of SAP97. So what we're pretty confident is going on here is that when this interaction between SAP97 and AMP receptors is disrupted either by getting rid of SAP97 or by getting rid of GluA1's PDZ binding domain, this causes this release of these sequestered pool of AMP receptors into the postsynaptic side of the synapses and these integrate. So then, of course, we were interested to know what is the impact of these schizophrenia-associated missense mutations that are found in SAP97's PDZ2 domain. This interaction between SAP97 and GLUA1's PDZ binding domain is very well established. Protein structure for this interaction has been resolved, which allowed us to perform protein structural modeling, allowing us to predict the impact that these mutations would have on this interaction. And our modeling did predict that they would be detrimental, but of course we wanted to validate these predictions, and so we did this by performing co-immunoprecipitation experiments where we find that indeed these schizophrenia-associated missense mutations in SAP97's PDZ2 domain significantly decreases the ability of SAP97 to bind to the GLUA1 subunit. So then again, we performed molecular replacement experiments. And so as I showed you before, if you eliminate SAP97 and dentate granule neurons, you see this very large increase in synaptic AMP receptor current amplitude. We can put back recombinant wild type SAP97 and restore normal synaptic AMP receptor function. But when we instead put back recombinant SAP97 that harbors these schizophrenia-associated missense mutations, we again see these large increases, likely pathological increases in glutamatergic synaptic strength in these neurons. So again, what we think is going on here is that when SAP97 is reduced, expression is reduced, as you see in individuals with 3q29 microdeletion mutations, or when this interaction is compromised via schizophrenia-associated missense mutations in SAP97's PDZ2 domain. Again, in both of these cases, you'll more than likely end up with this pathological increase in synaptic AMP receptor expression. So then another question that we were certainly interested in is whether or not SAP97 dysfunction, specifically within the dentate gyrus, whether or not this would be sufficient to produce schizophrenia-associated behavioral phenotypes. In rodents, I will be the first to admit that modeling schizophrenia in a rodent is an incredibly difficult task, but we had to start somewhere. And where we decided to start was looking at contextual episodic memory in rodents. And so there's an extensive literature documenting deficits in contextual information processing in individuals with schizophrenia. There's so much data, in fact, that there's even a contextual information theory of schizophrenia. So what we did was to take a virus that expresses our RNAi against SAP97, and we injected this virus directly into the dentate gyrus bilaterally in rats, and then looked at the performance of these animals on a novel object and context paradigm. And what we found was that when we locally reduce SAP97 expression within the dentate gyrus, we see a pretty substantial reduction in the performance of these animals on this novel object and context paradigm, demonstrating that contextual information processing is really compromised in these animals. We wanted to do our due diligence here, and we also injected this SAP97 RNAi virus into the CA1 region of the hippocampus, where we did not observe any changes in glutamatergic synapse function when we reduced SAP97 expression. We looked at the performance of rats on the same novel objects and context paradigm, and as we would have predicted, we see no deficit in contextual information processing. And so this suggests that compromising SAP97 functions locally within the dentate gyrus is sufficient to produce behavioral alterations, not unlike what you see in individuals with schizophrenia. So I would say that one of the more important questions sort of born out of this work is whether or not augmentation of glutamatergic synapse strength in dentate granule neurons, whether or not this represents a common feature in schizophrenia development. Because it could be possible that the synaptic pathology that we observe is specific only to individuals that have compromised SAP97 function. But in order to determine whether or not this synaptic pathology might be similar across more cases with schizophrenia, what we are very interested in doing is looking at these other genetic perturbations that have achieved whole genome significance for the disorder, compromise their function in dentate granule neurons, and determine whether or not we see a similar synaptic pathology. And so that's what we've started doing. We have a lot more to do, but basically we just went to our shelves and grabbed reagents that we had on hand to look at the impact of dysfunction of some of these other genes. The first one we looked at was GRIN2A. So GRIN2A is the gene that encodes the NMDA receptor subunit GLUN2A. It's actually been shown that when you reduce GLUN2A expression in neurons, that this produces, for reasons that aren't entirely clear to us, produced an increase in synaptic AMP receptor current amplitude, was shown by my good friend John Gray, who's now at UC Davis. And so we used a CRISPR against GRIN2A and transfected our dentate granule neurons with this CRISPR construct, and found that inhibiting GLUN2A expression in these neurons produces a very similar synaptic phenotype. It's increase in synaptic AMP receptor current amplitude. We've also looked at TRIO, which is another gene, synaptic regulatory protein, that my lab has been interested in for quite some time, and have a very well-established RNAi against TRIO, and express that in dentate granule neurons, and we see the exact same thing. So of the 3 schizophrenia-linked genes that we've looked at so far, they all seem to produce the same glutamatergic synapse strengthening in these neurons, which suggests that this particular synaptic pathology might be a common mechanism that produces increased glutamatergic neurotransmission in the hippocampus that you see in the early stages in schizophrenia development in humans. So with that, I will conclude. So what I've tried to convince you is that SAP97 prevents an extrasynaptic or perisynaptic pool of AMP receptors from reaching glutamatergic synapses and dentate granule neurons. The schizophrenia-related mutations compromise SAP97's ability to maintain this pool of AMP receptors, and results in what would appear to be a pathological elevation in glutamatergic synapse strength in the dentate gyrus. And that pathological synapse strengthening, specifically within the dentate gyrus, caused by schizophrenia-related SAP97 dysfunction, is likely sufficient to produce at least some symptoms associated with the disorder in rodents. And finally, what we think we've done here is to discover a specific synaptic pathology at a specific synapse in a specific brain region that's likely capable of driving the elevated hippocampal activity that's seen during the prodromal period of schizophrenia development in humans. And so with that, I will thank my laboratory. In particular, I want to thank Uni K, who's an incredibly talented graduate student in my laboratory who did the bulk of this work. Also thank Chin Tian, who's also an exceptionally talented graduate student in my lab, who helped with the project. I want to thank Scott Kanowski's laboratory, or collaborator, that his graduate student, Linda Tsan, who performed the behavioral experiments for us, and also Seva Katris's lab and his postdoc Anastasia, who did our protein modeling, and my funding sources. So yeah, with that, I'm happy to take any questions you all may have. Thank you for your attention.
Thanks a lot, Bruce. Awesome talk.
Hi, Bruce. I have a question for you.
Sure.
Okay. Good talk. So many people believe that schizophrenia is a neurodevelopmental disorder. But all your data here showed is that the SAP97 seems only affect neurotransmission. Is there any effects on the neurocircuitry formation, such as spine formation, or only neurodevelopmental effects?
So I mean, we have not looked at that specifically. It'd be very interesting to look at whether or not numbers of neurons change. These are certainly things that we want to get into. What you know, what I will say is what you see, there's something that you kind of see in the developmental expression pattern of a lot of genes that have achieved whole genome significance for the disorder is a developmental expression pattern that you have relatively low protein expression that does seem to peak in around adolescence. So SAP97 is this way. There's very little SAP97 expression in the brain early in postnatal development, and it does seem to peak sort of around adolescence. And GLUN2A, which has been implicated in schizophrenia for so long, and an MDA receptor inhibition is basically what allows the development of the glutamate theory of schizophrenia. GLUN2A expression is very low when we're born and peaks around adolescence. So, you know, I think there is some evidence that there is components to the disorder that likely come online around adolescence when you start to see symptom development. But again, there could certainly be other factors that trigger sort of a foundation that's required for the disease to ultimately manifest later in development around adolescence. But yeah, again, these are all questions that we are interested in. It's a very multifactorial disease, and we're just trying to pick out the pieces that may be important.
Okay, thank you.
Can I ask a question?
Sure.
Hello? Yeah, I have a naive question. How important is glutamate in schizophrenia? Specifically, would the glutamatergic antagonists work or help?
Yeah, so that's a huge question that we're interested in, and you know, I am trying to familiarize myself with this literature as much as I can. I'm interested in drug development on some level. The glutamate theory of schizophrenia has been around for a number of years, and some attempts to try to correct alterations in glutamatergic transmission have been made. What people have done primarily so far is to block class 2 metabotropic glutamate receptors, or sorry, not block them. These are PAMs. These are positive allosteric modulators, class 2 metabotropic glutamate receptors. And when you activate these receptors, this causes a reduction in presynaptic glutamate release. These seem to have antipsychotic properties. But I guess what my lab is most interested in this point is developing more targeted therapeutics that, because we see there's such regional specific alterations in glutamatergic synapse function in the brain, dentate gyrus sort of being one of them, we're very interested to see if we can't come up with therapeutic strategies that would allow us to dampen glutamatergic function, glutamatergic neurotransmission, and that's more localized. So that's what we're trying to do.
Okay, thank you.
Hey, awesome. Yeah.
Hi. Yeah, thanks. That's a great talk. A couple sort of questions. One is, yeah, so the dentate data is fascinating with SAP97, but of course, there's a couple other regions, you know, certainly they're very implicated. Have you looked at those physiologically, or with imaging? And also the other interesting gene is, of course, GRIA3 on your list, given you're looking at AMPA. So yeah, either of those. Yeah,
well, that's an exceptional question, and that's, now that we've identified this synaptic pathology in the dentate gyrus, schizophrenia is not all within the dentate gyrus. I would never here and try to convince you of that. But we are interested in this idea that we are not the first to propose, and that there seems to be on some level this synaptic pathology within the hippocampus that may sort of emerge and spread to other regions of the brain, including the prefrontal cortex. And so there are a number of questions that we're interested in at this point. One is if we produce this synaptic pathology locally within the dentate gyrus, and then look at neuronal activity in the prefrontal cortex, for example. Can we detect alterations in neuronal activity that grow from the synaptic pathology in the dentate gyrus? Another big question that we're interested in is SAP97 in the prefrontal cortex and other regions that have been heavily implicated in the disorder. It's sort of very interesting that we see this very specialized form of SAP97 that makes it out into the dendrites of these dentate granule neurons. We now think we have an antibody that targets that isoform specifically, the isoform of SAP97 that can make it into the dendrites. And so we're very interested in going to other parts of the brain and determining whether or not we have dendritically or synaptically localized SAP97 in these other regions. But yeah, very good questions. It's early days, but yeah. And yeah, GluA3. That's a big mystery to me. In the work that I'm aware of, in CA1 pyramidal neurons, when you eliminate GluA3 expression, the impact on synaptic function is very, very minimal. But I would love to be able to identify synapses where GluA3 is playing a major role. I've scoured the literature trying to get a toehold in a place to look, and I have yet to find anything compelling enough. But I am open to suggestion. I have had a number of people approach me like, we need to figure out what's going on with GluA3, and I could not agree more. Yeah, big open question.
All right, thanks. Thank you.
Kaiwen?
Hey, Bruce. Very nice talk. So I have a question in line with the previous one. So, so what makes the dentate granule cells so different from the other cell types in the hippocampus and other part of the brain? Is that the SAP97 only the knockdown in SAP97? I'm not very sure. You mentioned the other couple lines at the end of the talk that also shows the dentate granule cells have augmented synaptic transmissions. I'm not sure whether that is also limited to the dentate granule cells. My question is, what makes the dentate granule cells so different from like the CA1 pyramidal neurons that is vulnerable
in terms of what allows SAP97 to do what it does in those specific neurons?
Yes.
Yeah, yes. I mean, that's the question that we are also very interested in. I think we've made some headway there. SAP97 is subject to quite a bit of alternative splicing. So there are a number of different isoforms of SAP97. SAP97 that has the I3 splice insertion, that seems to be the SAP97 that is able to make it into the dendrites. When I told you that SAP97 was in the cell bodies of CA1 pyramidal neurons, but not in the dendrites, that is because they have an I2 splice insertion and not an I3 splice insertion. And it's actually nuclear. And this is consistent with some older work that was done in epithelial cells. What it's doing in the nuclei of these neurons, I have no idea. It might be a holdover from neuronal differentiation. What I can tell you is that when you get rid, our RNAi hits that isoform of SAP97 and has no effect on excitatory or inhibitory neurotransmission. So it's not doing anything to synapses. But yeah,
I see.
But yeah, it's alternative splicing that allows SAP97 to make it to the dendrites of these dentate granules.
So are the other two lines that you mentioned at the end of the talk which also have dentate granule cell defects? These phenotypes are also limited to dentate granule cells, or
actually don't know. I mean, gluin2A actually seems to be able to produce this pathological strengthening not only in dentate granule neurons but in CA1 pyramidal neurons as well. So in that case, it's not limited to it.
Okay, I see.
Yeah, so trio is actually kind of weird. We didn't expect that one to work. When we knock down trio and CA1 pyramidal neurons, we see a reduction in glutamatergic synapse function. When we do it dentate granule neurons, it's the opposite. I have some ideas for why that might be. There's a very interesting GEF environment in dentate granule neurons, but yeah, that was a bit shocking.
Okay, good. Thank you. Thanks.
Hey, Bruce,
Hi.
can I ask some question? Yeah,
Of course.
nice talk. Really interesting.
Thanks.
So the first question is that have you tried? Did you try like to knock out SAP97 like after the mouse was like mature already to see like is it a developmental or it's also impacts the mature?
Well, so
neuron
these are cultured slices, and so we wait for a period of time and then knock it down. So we knock it down. I guess what's the equivalent of P8, and then look at
I see. Yeah,
P14.
I know that system.
We've also done in utero electroporation experiments where we've knocked down, looked at acute slices. But yeah, that's that you've lost.
But you haven't tried like after like 8 weeks.
The answer to your question is no. I have not gone in to an adult mouse and gotten rid of SAP97.
Yeah, I'm just curious about this developmental feature, right? Yeah.
It could be that is something that's a very good question. Do you need to inhibit its expression very early on in development in order to see this synaptic phenotype? Yeah.
Okay, my next question is that so you see like the major changes like in APA receptor mediated current, right? The MDA is not that big change. So how do you feel like, because this like when people talk about like the glutamatergic hypothesis for schizophrenia, I mean, seems like most of the evidence come from the pharmacological evidence come from the MDA, but I mean the risk factors also include like APA receptors, right? Was it was like non-MD receptors also risk factors for the schizophrenia?
Yes,
and
so
what do you
GLUN2A is certainly a yeah. So if you have loss
that's
of
GLUN2A is an MD receptor, but
NMDA
there's other glutamate receptors involved. And what do you say is majorly is APA receptor mediated current change, right? And MDA is not changed much, right? Is it?
Exactly. No, we don't see much change in
So how do you feel like? Is it? I mean, my feeling is like, is it because that there are more pharmacological tools for MD receptor? That's why you see it mainly from MD receptors, or like APA receptor is also involved. You know, just you don't have tools to inhibit it easily.
So I think what it boils down to is local elevations and firing in the hippocampus during critical periods of development that are causing problems here. I think there are a number of ways that this abnormal firing or increased levels of activity in the hippocampus can come about. One is through aberrant strengthening of glutamatergic synapses onto dentate granule neurons, that being a seed. The pharmacological evidence that's been used, that what the glutamatergic theory of schizophrenia was born from, is ketamine. You put in ketamine, which inhibits NMDA receptors, and the model that's proposed is that for reasons that aren't exactly clear, seems to preferentially inhibit NMDA receptors that are on the excitatory synapses onto inhibitory neurons. And so that's supposed to shut down or reduce inhibition within the hippocampus. Is that what's happening?
So you feel is like whatever caused the hyperexcitation in hippocampus will lead to like schizophrenia-like stuff, right? NMDA is just part of it. I mean, other things could also do the same thing, right?
That's where I think things converge, and I think there are different ways you can get there, and that's what we're kind of trying to identify.
Cool. I'm working on 2A.
Oh yeah.
We can talk later. Yeah.
You want to work on 2A together?
I mean, I always work on MD receptors. All right. Talk to you later. Yeah.
We should chat.
Cool.
Right. Thanks so much, Bruce. And one fun thing is, I had a paper a few months ago where it wasn't the main point of the paper. We found the trio paralog caliran as a splicing target for this ALS protein. So I've been sort of intrigued by the idea that maybe similar mechanisms in schizophrenia and ALS could converge on some of the same targets. Anyway, we can talk
I
more.
know quite a bit about clearing. I've been studying that for a long time, so I'm happy to talk if you want.
Yeah, there's a cryptic exon that doesn't get included, but then in brains of patients with ALS, this cryptic exon gets inserted and it destabilizes the message, and there's proteins abolished.
Let's
Yeah, cool. So awesome talk. Great talk also, Giordano. Thanks seeing everyone. Don't forget, next week we'll take off because of the SFN meeting, but then we'll be right back at it the following week. See everyone soon.
Thank you very much.
Bye.
Great talk.
Thanks, guys.